Good afternoon to all of you. We are today here in our booth on the poultry science. And unfortunately, we are not together to shake hands and talk more closely, but we are here today with Professor Ferket, Dr. Ricardo Hayashi, and Dr. Cristiano Bortoluzzi. So Professor Ferket works at the North Carolina State University and is a nutritionist in work a lot of gut health. Dr. Ricardo Hayashi have done his doctor in microbiota as well, and Cristiano it's working now gut health and microbiota. So now we have here the USDA representative, that is Cristiano, is a postdoc there now, and Ricardo is from BRF. Thank you so much for all of you today to be here with me.
Thank you, Elizabeth. It's my pleasure to be here.
My pleasure. Thank you.
So it's just a starting process because microbiota is a subject that we would like to talk a lot in the next month. We're gonna have all week talking about microbiota in our webinar. We start with a more holistic way of microbiota with a professor of the Paris Musée Natural of Paris. Then you have a Professor Ferket here with a long time more for talk a little bit more, and you have Ricardo, and also we have Professor Chembo from Manitoba University to talk about swine microbiota. So for start to our talking today, I would like to say to everybody that is attending that you can send your questions to us, and we are able to see your questions here. And we start with Professor Ferket. Can you, for us to start as a professor, to describe what influenced the ecosystem of the enteric ecosystem in the animals?
Well, thank you, Elizabeth, for first of all inviting me to this webinar. This is something that we've been doing a lot here lately, presenting. All of our classes are taught this way now, and we're doing a lot of seminars. So this is becoming a very common way of communicating. First of all, I want to talk a little bit about the microflora, and that the gut of the chicken is quite a unique environment, right from the point where the bird consumes something in the oral cavity all the way through to the cloaca. And that there's a different microfloral ecosystem in each of these sections, and that microfloral milieu or ecosystem is influenced by digestive pH, osmotic properties, anaerobic and aerobic conditions can change. Gut motility and gizzard function has a significant impact on the structure and these sections within the gut, these separate little ecosystems. And of course, the nutrient composition, because they provide the nutrients that microflora then uses to ferment and influence the animal. So that gut microflora, everyone to remind, is pretty complex. Within the lumen of the gut, they provide a protective function, because if there are pathogens there, some what I would call good bacteria can displace these pathogens. They could produce antimicrobial factors that can influence that microbiota. They have an immune function, and they develop and they can influence the immunity of the host. They have a metabolic function where they can take dietary components and convert them into other kinds of nutrients that could be used by the microflora or even by the host. And even the host is producing secretions that feed these different organisms. And if you've got inflammation, it tends to feed more of the pathogenic type organisms. But if you've got a very well-structured, healthy gut brush border, then it could also, the secretions of mucin, for example, could then feed good bacteria as well. So, Elizabeth, when we think about the gut, we have to think of it much like an ecosystem. And we're all familiar with ecosystems, whether it's a pond or a forest or a field. But if you think about the gut, it is its own unique ecosystem. And all ecosystems have some properties that can influence how that ecosystem develops and maintains. So the first one is you want to establish an ecological environment to cultivate it, and it's influenced by its physical structure. And then it also has certain symbiotic microflora that thrive in a balanced manner in that ecosystem, and it requires you to seed the right type of organisms there right from the beginning. The next is just the nutrient balance. So the idea there is that you want to feed the symbiotic organisms and starve the pathogens. And so there are a variety of different methods that we could talk about and how to influence that nutrient balance for that ecosystem. And finally is maintaining that symbiotic stability of that microflora. And that can be done by either controlling the diversity and the microbial load within that ecosystem, and then also how these organisms not only compete with one another, but there are techniques where you can weed out the bad pathogens and leaving and preserving the good ones that are more symbiotic.
So this means that there is a lot of factors that affecting the what we call ecosystem. And maybe it will be nice if I repeat the questions for Cristiano that I did a few weeks ago about the concept of microbiota, microbiome, and ecosystem. How can we define it? Because still there's people that call microflora. Can you talk a little bit about that again, Cristiano?
Yeah, sure. Well, thank you, Elizabeth, for the invitation to be here. It's my pleasure again to collaborate. And yes, I think when we talk about these different terms, I think we need to have that very clear, right, to avoid any confusion. And if you guys watched yesterday's symposium on bioinformatics, the first speaker talked about microflora. Sometimes we use microflora, but it's not a more accurate term to talk. And she said, well, the microflora, they are not small trees in the gut or small plants in the gut. So I think the more accurate term is to use microbiota, right? And microbiota would be more like when we are studying a population, like the bacteria population or the virus or the fungi population in the gut. When you talk about microbiome, we are talking more about besides the population, the bacteria, we are also talking about maybe some metabolites that are being produced by those bacteria, the metabolites that are present in the gut that are influencing other groups of bacteria, as Professor Ferket said, or they are influencing the host. But also the metagenome, when we talk about the full potential of that microbiome, and then instead of using like small fragments, sequencing small fragments as we do like the 16S, when you talk about metagenome, we are talking about sequencing the whole genome of the bacteria, so we can know we will be more clear what is the genetic potential of that the population. I think, yeah, we need to have these terms very clear to avoid any confusion. And well, I agree with what Professor Ferket said. Sometimes I'm biased to talk about microbiota because I'm a big believer of the microbiota. Sometimes it's hard to study and to understand, to interpret the data that we get, but I think we are on the right way with all of these methodologies and new technologies that are being developed to help us to understand more about these concepts, microbiota, microbiome, and metagenome.
Thank you, Cristiano. And now, Dr. Ricardo, it's a pleasure to have you here. Thank you for your time. And what I'm wondering here, and still again, people that are watching us, just send your questions and we can address your questions for all of the speakers here. But Ricardo, I was thinking about you went from the academy to the industry now, so you are representative of industry. How is the vision, like the concept that Dr. Ferket and Dr. Bortoluzzi share with us in the industry? How do you guys are starting to apply this concept or thinking about this concept in the practical way?
Okay, thank you, Elizabeth, for the invite. It's a big pleasure to be here in this discussion with great professionals about this topic. Well, about microbiome, it's something that I'm really passionate, particularly because I've been studying this topic since my PhD a couple of years ago. And I went to industry, now in BRF, Brazil Foods. And I'm bringing this topic because I think gut microbiota is something very important. I think it's the new center of everything about all ecosystems. It's a kind of a bridge, a correlation between everything, between the environment, the diet, the host, the genetics, and what's going on around the host. So I think it's a very important topic, and you should get more data and more information and more knowledge about this topic. My challenge now is how to bring these concepts that I've learned in the academy to industry, because there are two different scenarios. And I think the first one is try to solve the main problems that we have in the field. What are them? Like, for example, we have some units that are passing through problems with initial mortality, or some specific chronic problems about feed conversion and the results related to performance. So we have a lot of challenges that we cannot find the answers. And I think if we look more to the gut microbiome in general, in the correct way, I think we can get more information and more data about this. When I say about look to the right way, I think the first one challenge that we are passing through, we have seen a lot of published papers analyzing in different ways about gut microbiome. There is no like a standard to analyze the gut microbiome in my point of view. I may be wrong, but I think there's a lot of mistakes that companies and researchers, universities, that are doing now about this topic, how to analyze the gut microbiome and compare scenarios, compare when you have like a challenge, a specific challenge and other stuff. So I think the first step is get more data, more information, more database. It's really important to create some profiles, some patterns, and then correlate with some specific scenarios that I mentioned before. The real demands that we are passing through the field. So I think there's a lot of challenge about this topic, and I really like this subject, and I think we have a lot of challenges to discover and get more information about it.
So thank you for your answer, but this is also brings something. For example, in human side, there is a company that I can even talk the name, that is Bioma, for example, that you buy the kit and then you go home and then you send the sample for them. And at the beginning, the price, it was very expensive, and in fact, they start to building a platform, maybe like they started, I think 2-3 years ago. So now they have a platform with some informations that make them to give some advice that, because at the beginning, when I first saw what they say to people, it was very general, like eat more vegetables or things like that. So I will bring these questions for us to think in poultry science and poultry science and poultry productions. How will be the best way for us to building a platform that allows us to understand, like Professor Ferket say, oh, we have many factors like nutrition factors and environment and that influence this ecosystem. So my question for all of you, then each one can answer in. It's how do you think that will be the future for the microbiota studies? How the industry, the science could work together in a platform for us to have the answer for what is the bad, what is the not bad microbiota. We can start with Professor Ferket because of the.
Well, that's a very good question, Elizabeth. And I'm on a committee called the National Animal Nutrition Program. There is a website. So if you just Google National Animal Nutrition Program, that is a large database website that includes ingredient analysis and also modeling. And part of our initiative there is actually to create a large depository of data that's related to nutrition. And there you could upload your formulas, growth performance, big data of microbiome data, just the raw data, as long as you can describe that. And that's important because we need lots of data, but with our availability of machine learning and artificial intelligence software, we can now use that data to actually try to discover more about what the microbiome is about. It's very complex, just as Ricardo kind of mentioned and that when we look at the data, I mean, it's difficult to identify patterns without very sophisticated tools. And so those tools are now becoming more available. We're starting to learn how to use that. We will have algorithms published on that website that allows you to use for big data analytics, particularly related to the microbiome. I just got to remind everybody that it's not just the bacteria that live in that ecosystem, it's what they do. And that's really what's influenced there. It's not necessarily particular species and phyla. It's what's the whole microbiome function. What is their metabolism that is contributing to the gut health issue? So those are the things that we really need to have a better handle on. And I think the National Animal Nutrition Program website and programs like that really contribute to that.
Cristiano, what's your opinion? How can you build in this platform? How is our position and influence to have a better data in the future?
Yeah, I think I agree with what Dr. Ferket and Dr. Ricardo said. I think we need to have a much better standardization in like starting even with the sampling. When we are going to sample the birds, we need to try to, my opinion is that we need to try to reduce the bird-to-bird variability. Instead of like collecting one bird, try to make a pool of 3 or 4 birds to try to reduce that variability. And I think that will give us a much stronger information what's going on with the treatment or with the flock. And after that, we need to have a better standardization of the protocols that we use for DNA isolation and then for sequencing. What platform we are going to use for sequencing, and also we have that. I think we need to have like a common ground when we talk about research, like different results. We are not someone used one, let's say one protocol for DNA isolation. I am using another one, so that is already something that it can vary and can change the results. And after that, like the sequencing, the protocols that are being used for sequencing, and then the softwares that are being used for to analyze the data, the bioinformatic, and then the database that I'm comparing my sequences to. I think all of that we need, as the speaker talked about yesterday, we need to have a better standardization from the beginning to the end. It includes like the sample collection until like data analysis. And I think we need to build this same ground when we talk about that.
Ricardo, for just to, it's the same subject, but just in your case, I one day, it's for all of you, but I address just to Ricardo because of the order. I have been in a conference in London, and they talk about microbiota, and I also say why we don't do a standard, like why we are talking. And then one of the panelists say to me, oh, but because it's a new science area, if you put a standard, maybe we are like reducing the science to discover new methodologies and new ways to look at. Do you agree with it, Ricardo, that we should think about the standard, but at some time, maybe the standard that you have today, like if you compare what we start to study microbiota years ago and now, if you just standardize, you are not block the possibility to grow more in terms of methodologies?
Well, Elizabeth, that's a good question, because somehow I agree with his point of view, because I think science should be like, we need to think out of the box all the time. We need to create different options and opportunities all the time. But scientific method is just only one, I think. So we need to follow, I think, the best one, like method, like Cristiano said, about how we do the extractions, about the sampling and the method, the sequencing method and everything. I think we should make a pattern about this. What we need to do differently, I think, when we talk about gut microbiome, when we go to the field, for example, we see different scenarios, even in the same company. For example, in BRIF, we have huge different scenarios all the time. We have different units, different places, different housings, different structures, becoming from different companies in old times. Now it's the same one. So we need to make a standard and see how the microbiota it's been modulating with these different scenarios. So now we are in just phase one, trying to create these profiles and see and correlate with good performances, bad performance, and chronic problems that we have facing, and stuff like that. So I think we need to think out of the box sometimes, but the scientific method should be followed all the time. I think this is universal, and
so do you think that, for example, we should have like the university and research centers looking for different ways, but the industry should have like a standard? Another day I was like talking to Dr. Peter Cristal and about another subject about amino acids, but he said to me, you know, I have to be consistent. So because I'm in the field, I need to have some consistency. So I understand that what you guys are saying, that we supposed to have more open mind to new methodologies that maybe we don't have now, but we can have in the future. But when you talk about practical way, we should standardize it, and maybe the Professor Ferket, do you think that poultry science, who could be the Poultry Science Association, who could be the lead of this big project for us to put out together this information, try to do something practical? Because you have been talking about microbiota at least in 10 years from now, that we are always talking a little bit and increasing. But if you think about maybe next poultry science, can you think about to have like a committee or something that really think about to put together these informations and have something practical?
I think that's an excellent idea to put on a symposium that talks about protocols of measuring microbiome. One thing that comes to my mind is, well, what sample do we take? The cecal sample, fecal sample, ileal sample, jejunum, and because each of those sections will have a uniquely different microbiome. So we need to have a standardized type of a method. I'm the director of the Animal Health and Nutrition Consortium. It's a new consortium. It's a partnership between industry and university. It's within our NC State University right now, but it has the capability of expanding beyond that. And a lot of the efforts associated with that consortium now is to develop tools and protocols that everybody could use and agree on for developing and assessing gut health or performance and using different kinds of tools. And so we're working on biomarkers, how to analyze microbiome, how to sample things, how to set up an experiment. These are things that everybody commonly is interested in. So I think certainly whether it's the consortium like the Animal Health and Nutrition Consortium that I direct at NC State, or at Poultry Science, or the National Animal Nutrition Program, there does need to have a centralized organization, I think, where we can then freely contribute to that pool of knowledge and allow to create a standard in sampling, etc. And of course, these things can all change, so it doesn't have to be a static procedure, but once we identify a standard, and you can compare it now to something else that you improve, and then possibly change that methodology for the better.
And Ricardo, for you, if for example there is some association or something, some starting or university or whatever, start this standardization, are you in the industry open to share results, or do you think that this is like a dream that companies are not ready to share the results, to discuss and to building a strong platform that we can rely to understand the future answer?
Well, BRF is working very hard to make like an open innovation system, which we can collaborate with researchers and universities, startups and other companies in order to get more information, to evaluate to evolve the industry, to get more information and solve our problems. So I think it's a very nice question, Elizabeth, because we are very open to make a system. We have a lot of great partners right now doing this project with us, involving gut microbiome, trying to identify these biomarkers, trying to design specific trials in the field that we can get information about our problems. And it's something very open to work in collaboration with everybody.
And Cristiano, I was thinking here about this. We talk a lot about intestinal microbiota, intestinal health microbiota, but lately, because of COVID especially, people are really relay also the respiratory microbiota as well as part of health, and the connections between the respiratory and the gut microbiota in the health of behavior and performance. How do you think about that? We need to change our protocols now to also contemplate the respiratory tract? How do you see it?
Well, I actually don't have much experience. Well, I never did any study looking at the respiratory tract microbiota, but I think that the respiratory tract is much more affected, maybe by the environment, I would think, but also by maybe that will be influenced also on the the intestinal microbiota with regards with the development of the immune system. I think all of this mucosa immunity, it's all interconnected. So I do believe that the changes that we make in the intestine, the microbiota, the role that microbiota, the intestinal microbiota is having, like modulating the immune system in the gut, may have influence on the microbiota in the development of the immune system in the respiratory tract. I saw the other day this review paper that came out like these last few months, talking about that relationship between gut microbiota and the susceptibility to respiratory diseases, of course, including the COVID-19. And many of the receptors, and something that I was reading, some of the receptors that are present in the respiratory tract are also present in the gut, in the enterocytes. So I do believe that the maturation and the modulation of the mucosal immune system in the gut will select or will influence the immune system in the respiratory tract, and also selecting different groups of microorganisms. But I think that is very speculative. I haven't seen at least studies on that, mainly in poultry. But I do believe that the influence on that is big.
Well, today people I think are very shy to ask a question. Normally I have a lot, but I received a question here from Guilherme Matuela, and it's so nice because he was my ex-students. However, there is no ex-students, right, Professor Ferket? So I was like here very happy today because I have a Professor Ferket that for me, however, we never officially worked together, was my advisor for many aspects of my life. We worked together in many situations and in different situations. And I have Ricardo here that have been also my students, and Cristiano that we start to working more lately. So for me, it's like generations that is going on in terms of nutrition and gut health, and I'm very proud of all this. So Guilherme are asking a very, very, very applied question, because we talk about microbiota. Another day I was talking with Billy Argus here, and Tainá was with us, like also a new generation of the students. And she said that the litter, it's a live organism inside the ecosystem of poultry productions. So Guilherme was asking us, all of you, that what's better between litter that was used or litter that was reused. And I think that this is give you a gap for you to talk a lot about this. So I just let the questions here and start with Professor Ferket, and then I go for all of you to answer Guilherme questions. And thank you, Guilherme, for staying with us and for the question.
Well, Guilherme, that's a good question, because the litter is a part of that whole ecosystem, and certainly there it is an indication of the gut microflora or the gut microbiome of each individual animal. It's a composite, but also through reverse peristalsis and what we call cecal drinking. I mean, when a chicken sits on the litter, it literally can pick up the microorganisms from the environment as well. So that litter can be a means of delivering a good balanced ecosystem, or it can be one that can create disease. And so I think that litter quality is absolutely important for the maintenance of the gut microflora. We know that when we got wet litter, that it promotes certain organisms to proliferate, and that these might potentially be pathogenic as well and competitive with the birds. And it not only affects the microflora within the intestinal tract, but also food safety issues related to what could the animal on the outside during processing or foot pad qualities. So certainly maintaining that litter quality. Here in the United States, it's pretty common to have multiple flocks on litter. And if you've got a good flock where you've got pretty good performance, guard that litter as if it is a prebiotic or a probiotic for the next flock. But if you've got a very poor health type of a flock where you've had many challenges, then consider either amending that litter significantly or cleaning it out and starting again. Always consider the quality of that litter as being an important component of a healthy gut microbiome.
Cristiano, what's your opinion?
Yes, well, I totally agree with Professor Ferket, and we talked about this the other day as well. And I remember when I was in vet school in Palotina at Federal University of Paraná, I was, maybe that was my third year in vet school, and there was this guy that came, he used to work for a poultry integrator in the region. And he said, do you guys know which one is the best leader? And we were, maybe wood shavings or something else. And he said, no, the better leader is the chicken poop, is the poop of the chickens. And we were like, that makes sense if it's if a good quality leader, well maintained, of course, like devoid of any pathogens. I think that plays a big role in the establishment and in the maintenance of the microbiota. And I think Ricardo can give us a much better idea, but regarding to the performance of the flocks. But as we talked the other day, and that's something I have learned, that the best the better performance, the best results in performance are obtained with maybe second, the third, or fourth cycle, not in the first. Instead, in the first, we may have the worst cases of outbreaks of necrotic enteritis or coccidiosis or these diseases. Maybe Ricardo can give us a better idea on that. But yeah, that good quality leader is a good source of probiotic bacteria for sure, and to establish and maintain the gut microbiome.
Yeah, Ricardo has I think the guy that more knows about the litter reuse, that is Marco Dalpra, it's his colleague. So I think I'm really looking for your answer, Ricardo, because you have a lot of information.
I'm very lucky. I'm very lucky to work with Marcos Dalpra. Marcos Dalpra was with you in a couple of weeks ago talking about litter management and the relation about animal health. And it's very clear to see the relation about the good procedures, good management of the litter with good results. In BRF, we are doing more than 10 to maybe 20 cycles above the same litter as well.
Wow.
So, and we go better results when we do the right management, when we do the right procedures in the downtime between flocks, the cleaning, the disinfection in the environment, the management of the litter, fermentation, and control of insects and everything. So when we do that, we can see very good results, cycle after cycle, and probably we're gonna have maybe a good fingerprints of the environment, the litter, related to the gut microbiome. This is one of the projects that we are designing right now, trying to correlate with this good or bad management of the litter, and trying to compare with the sequencing and the composition of the gut microbiome of these scenarios. I think we're gonna get very nice results about this project, but I'm pretty sure that it's very totally correlated with the good management and the age of the litter, because as Dr. Ferket said before, it's a kind of a probiotic in the environment, which is gonna help to colonize early the gut microbiota of the little chicks that are coming after housing. So I think this is a very known concept, very studied concept for Marcos da Praia and other researchers. This is very well known, and I'm pretty sure that we're gonna get more information correlated with the gut microbiome as well.
Yeah, I think litter is important. You see that I have the day that I have Marco da Praia, it was like no space more in the webinar because people are really interested in that. So I think that is really something practical that people like a lot. And I have also an honor to have Dr. Christy Schoengerti listen to us. Thank you, Christy, to be with us. And she comments that it's very nice to have the industry, the university, working and talking about the standardized microbiota. So, Professor Ferket, I think you have some mission here and lead this project maybe on. And I have also a question from Fernando Fontanilla. Thank you, Fernando, for staying with us from Argentina. And he asked us something that is very interesting because all the time, at least when I start, people say, oh, for broilers, for monogastric, we need to avoid any kind of fever because we have to have a very high digested diet. So he brings to you the questions that how your opinion about the fiber composition in the diet, and if how this could affect microbiota. So, Professor Ferket, can you start, and then we can.
Well, thank you for giving me the first crack at every question. Well, certainly, what you put in the diet can be, the food source. But what we often think about is just the total nutrients that's actually digested. But really, when we consider what's going on with the microbiota, it's what is not digested and used by the animal has the greatest impact on the gut microflora as well. So if you have poor digestible protein, poor digestible fat, poor digestible starches, etc., that will feed competitive organisms within the gut that may cause problems. However, if you're feeding and have nutrients that is not used normally by the animal, such as non-starch polysaccharides, certain kinds of fibers, they're not used by the animal, but then they are good food sources for the symbiotic type organisms. So certainly the type of fiber has a tremendous effect on other types of metabolites that these microbiota produce, like organic acids. They might produce vitamins. They can produce and make even minerals more bioavailable as well. And these are all absorbed very often in the hindgut. So certainly think about when you're formulating a diet is what is the non-digestible part of that diet? Is it something that would be food for symbiotic organisms, or is that undigestible component food for pathogens? And we have to consider that when we're formulating our diets.
Cristiano, do you have some?
No, yeah, I totally agree with Professor Ferket. I think he gave the best answer. All of these microbiota, mainly the cica, right? When these non-starch polysaccharides that reach the cica, they can be used, and then these molecules can be fermented by many types of bacteria, and they will produce these short-chain fatty acids that have a huge impact on the health of the host. Maybe some antimicrobial effect locally as well, and also will be absorbed and have systemic effects. So yeah, I think that is actually that is the concept of prebiotics, right? So actually those prebiotics are non-digestible particles that will feed the good bacteria. And of course, sometimes we don't want to formulate a diet. Many times we don't formulate a diet to feed the microorganism. We formulate a diet to feed the host and make the the broilers grow faster and be more efficient. But yeah, I think when we give those sources, those substrates to the bacteria, I think they can give back, and they will producing these molecules that have effect on the host. I have collaborated with some folks in Brazil doing a study on enzymes, and I think that the enzyme concept comes with that, because sometimes the enzyme can have an effect on degrading some of these fibers, and those particles can be used by the good bacteria. And actually, when they supplemented the diet with different types of enzymes, the profile of the microbiota changed. So probably we did not analyze the profile or the fiber in the digester, but for sure the profile of the fiber that reached the cica was different, and that had an effect on the microbiota, and probably that was one of the points that benefited made the broilers to grow faster.
Ricardo, are you doing some studies with fiber in the diet? Do you have some experience or share with us?
Not that much, Elizabeth, because I'm not a nutritionist, but I think knowing about a little bit about fiber, I think it's very important to see the behavior of the gut microbiota when you use different types and quality of fibers. So maybe you can make a different strategy when you have some different scenarios, and then you can complement, you can use some additive, you can use some specific type of enzyme, for example, try to compensate and get the most of it. I think it's a good insight to work with this.
I have one question from Jared Oxford, it's our new colleague in Jefo. So welcome, Jared, to our team. And I think it's for you, Ricardo, because he was asking us, for example, I think it's going on with the question of Guilherme about litter. And he said that if it will be not more interesting when you think about probiotic to put in the litter instead to put in the how can I say in the feed or in the water.
Well, this is very interesting, very important to discuss, because we have I have seen some projects going on to the environment, like environmental probiotics, which we are trying to get more information, but we have seen good results and good relation with the environment, not only to the environment, but the animal performance, and probably modulating the gut microbiome. So I think this is a new research line trying to understand what's going on about modulation of the environment, the air, the litter, and everything. And probably, I'm pretty sure that it's gonna make a good correlation with the gut microbiome. And we have seen good performance results as well, feed conversion, mortality, slaughterhouses results as well. So I think it is a very good research line. I haven't seen so many published papers about this area, but it's a very good topic to stimulate people to do more research about it.
A question here from my super friend Sandra Rodriguez. Hello, Sandra. It's about, can we, for all of you, can we apply like all the knowledge that you have for ruminant side, rumen, in microbiota? So it's basically what she's asking us. What do you think about to take advantage of studies in ruminants, including nutrition and the dynamic models to test different diets? I actually put some questions with her. Like for me, I know that when you talk about microbiota, there is interactions with the of course immune response and environment. But for example, Professor Ferket, in case of nutrients, I know that you have this experience that we can like regulate some nutrients and change the microbiota. Can you go for this model in vitro? So it's two questions in one. It's like, can we use the nutrition, the knowledge of ruminants for, and also the model with different models that they do in vitro with ruminants for study microbiota, and also the concept of make a diet for the microbiota instead that look at just the requirements of the host.
Yeah, this is a good question, because ruminants are foregut fermenters and pigs and chickens are hindgut fermenters. And yet we think about a monogastric animal as being a big tube, and we just want to make sure that nutrients are digestible and is available for the animal, and we don't care that much about what's left over. But the ruminant nutritionists and the ruminant physiologists have a lot of knowledge in terms of fermentation, and we know that nutrient availability, the type of fiber, the texture of the fiber, there's a lot of different nuances about that can influence the type of fermentation that's going on. One of the things that ruminants have been using is what is called bypass type technologies, where they will coat either an amino acid or coat proteins or do something where it actually slows these the release of these nutrients, so that competitive organisms within the rumen don't overpower, and it allows the good bacteria, the symbiotic ones, to proliferate. That same concept could be utilized in the monogastrics as well. For example, if you overfeed vitamins, for instance, you can actually feed pathogens, because these pathogens are competitive. They want those same vitamins. But then the fiber-producing bacteria or fiber-consuming bacteria, the symbiotic ones, generally produce vitamins. And if there is an oversupply of vitamins, and minerals, it actually could suppress the symbiotic bacteria and cause a proliferation of the more pathogenic type or competitive type organisms. We have recently done some research on vitamins, and in poultry diets, we tend to put in lots of vitamins. We formulate well over the requirements just for safety purposes. And vitamins occupy a very small part of the diet. Yes, per unit of vitamin premix, they're expensive, but in terms of the diet, they're not. So people tend to put way more vitamins in that diet. And overdoing vitamins may adversely affect the microbiota profile. We've done some research where we encapsulate that vitamin in a lipid coating, and it's a matrix that slows the release of the nutrients. And then by feeding lower levels of vitamins or slowing that release, tends to shift the gut microflora towards a more symbiotic profile, which benefits the animal. There are two things that happens when we use that, in that not only there's a cost savings possibly in the vitamin supplementation, but you can manipulate the organism or the microflora by how you time release these nutrients. So this same technology is used a lot in plant sciences, where they do slow release fertilizers and things like that, all having a beneficial effect of soil microbiota. So I think the same concept could be very useful, and learning from our friends who are the ruminant physiologists, and using models like a rumen model to actually test these different kinds of delivery mechanisms and formulation profiles.
So, Cristiano, do you think that ruminants could be taken account for us to think about microbiota?
Yeah, absolutely. I think agreeing with Professor Ferket, using this concept of microencapsulation to try to target some specific microorganisms in the lower gut, let's say in poultry, in the microbiota in the cica, and I have seen some studies with, let's say, vitamin B2 or B3 that can increase the proliferation of some butyrate-producing bacteria. So those vitamins can specifically be used by those species of bacteria that they do not have the capability to produce their own vitamins. They have to use the vitamins produced by other microorganisms or from what is in the diet, what is in the digesta. So I think using that concept of targeting some using this microencapsulation to target some specific microorganisms in the lower gut might have some benefits to the poultry. I haven't seen results in poultry with that. I have seen results in humans, and there is this article. Actually, they call when they are talking about, let's say, vitamin B2. And they call vitamin B2 a new prebiotic that can feed some specific groups of bacteria, of course, to get the benefits from those bacteria. And regarding the methodologies, when they use these, try to mimic the rooming fermentation in the lab, I think we can use that in poultry as well, try to mimic the conditions in the cica, in the lab, and to have more these in vitro results, how the different microorganisms from the cica can respond to different nutrients in the lab before applying in vivo animals. So maybe that is a possibility. Sorry, for poultry as well, that sometimes we don't take into consideration. It's good to use animals, and maybe poultry, it's cheap and easy to have, and to have a big number of animals and collect samples. But I think maybe using these in vitro type of studies will give us good answers as well.
Maybe as a concept, right? What do you think, Ricardo, about that? Do you I know that you are in the field, so you have how many animals you want to analyze, but how do you think about implying like new concepts of nutrition and field or another methodologies that you can bring from ruminant side, for example?
Well, I think I agree with everything that Dr. Ferket and Cristiano said about it. To adapt these in vitro methodologies, I think it's very important to use less animals as we possible. Like, I think we need to find some methods and analysis less invasive that we are doing right now. So I think this in vitro analysis in the lab, which we can simulate the behavior and the functions of the gut microbiota, looking for metabolites, for example, or something like that. I think it's very worthwhile to adapt and to validate this, because we are looking for like cheaper analysis, less invasive, to not kill birds anymore, because we are going to this way. And it's very important to adapt something that we can simulate in vitro instead doing in vivo and killing birds and breaking rules about the animal welfare and something like that. It's very important.
It's amazing. The time is passing so fast here that we are going to the end. So just before we finalize, I want each one of you have like a final message for the people that was watching us and listening to us today about the future of microbiota. And of course, I will start with Professor Ferket, as we have this model.
Well, thank you, Elizabeth, for this opportunity to share with everyone. And yes, this is a new area. I think, as we discussed, understanding the microbiota, the microbial ecosystem of the intestinal tract has created a revival in research. It allows us to now take all the disciplines of nutrition and physiology and immunology and management, and it helps us now to communicate and in a way that is relevant for our animal production. Of course, studying in this has a One World Health mission as well, because it's important for the health of our own systems. We're beginning to learn so much that the microbiome and the microbiota within the intestinal tract is it's the next organ that we didn't realize. There are more genes being expressed in the microbiota than all the genes within the animal, way more cells within the gut than even the animal itself. So as a nutritionist, I start thinking, am I feeding an animal or am I feeding that microbiome? Am I feeding the organisms? And it's really the holistic approach to animal production. Once we understand, and we may be a long ways from fully understanding that world called the microbiota within the intestinal tract, but certainly as we begin to think in those terms, think in terms of managing the enteric ecosystem, we are far better to solve solutions that plague us in terms of animal performance, gut health. Certainly in these days where we have to stop using antimicrobial agents, we've got issues related to resistance, and we have a multiple of other kinds of problems that we haven't even begun to talk about, like viruses and the interaction of what could happen within the microbiota. So thank you so much, Elizabeth, for bringing us all together to discuss this very important topic.
Thank you. Cristiano?
Yeah, well, thank you so much, Elizabeth, once again for this opportunity to come and talk about this topic. As I said, I love studying the microbiota. And sometimes it's frustrating when we don't see what we were expecting to see, but I think that cannot stop us to do a good science and do good research and keep looking for the answers we have. And I think the microbiome, as we discussed, is very easy to manipulate, and the answers that we get may change a lot. But I think we are on the way with all of these new methodologies being developed to understand of the microbiota, the composition, and also what the microbiota is doing. And I think it is, and it will be a big player when we talk about non-antibiotic production, how we can manipulate the microbiota to try to minimize all of the diseases or the proliferation of pathogens in the gut. So I think it's a big player. It's a therapeutic target that we need to always think about, and never forget about the immune system. Also, the role that the microbiota, or the relationship between the microbiota and the immune system, because they are working together, the immune system and the microbiota, one influencing the other, and of course all of the nutrition aspect on that. Well, thank you so much, Elizabeth, once again, and I will be happy to talk even further about all of these topics.
For sure. Immunity, you are in the right place. Like you and Michael Good, for sure, you will do a good work in this area, microbiota and immunity. And Ricardo, it's your turn.
Thank you very much for being here. Once again, it's a big pleasure to discuss a little bit more about this topic. And the more I read, the more I studied about this topic, the less I know in the end, because we're trying to get more information, and in the end, we have more questions about it. And I mean, this is very nice about the gut microbiota. This is science. I think we should bring this concept closer to the industry. It's very important, to have these events, these discussions, these open discussions about these topics. This is something that it's not like street secret anymore. This is something that we can discuss about it. This is modern industry. To share what we are doing, to know what Dr. Peter Ferket, Dr. Cristiano Bortoluzzi is doing in USDA, in Jefo, in other companies. This is like we have to learn together to bring everybody to the same ground, to the same level, and to evolve about this topic. When I talk about gut microbiome, I cannot do anything by myself, because for doing like a complex projects involving gut microbiome, I need to have good statistic people. I have we need to have like people from IT, from bioinformatics, people from the field, veterinarians, lab technicians. I mean, this is very like a multiple types of people, professionals doing the same projects. So we need to encourage these projects to get more attention for other companies, for academy, for researchers, labs, and everybody to put the same, to put everybody in the same ground to get more information. This is very important. And thank you for being here again. It's a big pleasure to discuss it with you.
Thank you so much for all of you. Thank you for your time today. I can say that it's a happy day. You know, a happy day that I have a presence of you, for me, my professor, my students, my colleagues today, and stay in Jefo. So it's a very happy day with all these people listening to us. It's really, I'm very happy. Thank you so much for your time, and I hope to have you guys very soon in the next webinar. For sure, I'll have some of you, but I hope that the world start to become more easy for us to connect it personally, because as Brazilian, I miss the hug, I miss the true contact. Thank you so much. Thank you for everybody that stay at home, and tomorrow we'll be here again in Spanish to talk about immunity, nutrition. It will be a nice day as well. Thank you so much for all of you.
Thank you.
Thank you.
Bye.
Bye-bye.
Bye-bye.